Supporting information, S1 video, Bird of Paradise behavioral scoring demonstration. In this video, a male western Parotia selvata performs a species-typical courtship dance for females perching above. This video demonstrates the concordance between a subsample of our scored behaviors and the actual performance of the bird in real time. The behaviors represent BP1, BP2, SS1, O3, OPMH, OPMF, OPMAH, and OPAC1. Users who cannot download the video can also view it here. Link: BP1, body position moving, BP2, changing direction while moving, OPAC1, ornamental flank plumage accentuation by moving the torso, OPAH, ornamental head plumage accentuation by moving the head, OPMF, ornamental flank plumage accentuation by moving those feathers, OPMH, ornamental head plumage accentuation by moving those feathers, O3 bowing, SS1 shape-shifting, S1 table, MPGLS analyses of communication-relevant influences on 3 axes of courtship phenotype diversity. Categorical comparisons of display site are made with respect to ground-displaying birds, and breeding system comparisons are made with respect to solitarily displaying birds, MPGLS, multiple phylogenetic generalized least squares. S2 table, MPGLS analyses of communication-relevant influences on 3 axes of courtship phenotypic richness. Categorical comparisons of display site are made with respect to ground-displaying birds, and breeding system comparisons are made with respect to solitarily displaying birds. S5 table, MPGLS analyses of communication-relevant influences on 3 axes of courtship phenotype diversity conducted using behavioral complexity metrics from a 10-second and a 60-second time window. For comparison, the analyses presented in the main text focus on behavioral complexity estimated from a 50-second time window. S9 table, partial summary PC1 to PC3 of PCA of 5,739 notes produced by 32 birds of paradise species. PC loadings for PC1 to PC3 were used to plot notes in 3-dimensional PCA space prior to agglomerative hierarchical clustering based on Euclidean distances to categorize notes. S10 table, MPGLS analyses of communication-relevant influences on 3 axes of courtship phenotype diversity conducted only on species without imputed species-level values. S1 figure, there is no evidence for correlated evolution between color and behavioral diversity among birds of paradise. MPGLS regression reveals no significant relationship between behavioral and color diversity when controlling for acoustic diversity, display height, and display proximity. This plot is a phylosignal space plot in which species ornamentation values are plotted with colored circles corresponding to display environment and mating system and are connected based on their phylogenetic relationships. Species locations represent tip values for log-transformed behavioral and color diversity. S2 figure, accumulation of unique behaviors plateaus by time windows of approximately 50 seconds for most species. Note, these are unique behaviors per individual, not per clip. This distinction is important because some individuals were recorded in several clips, but in the longer clips, they might not be doing much behaviorally, leading to the initially surprising drop in unique behaviors at certain longer window sizes. S3 figure, pairwise comparisons of behavioral richness, number of unique behaviors, estimates for windows between 10 and 60 seconds in duration. S5 figure, dorsal view of raw left side and clustered right side images taken of a Wilson's bird of paradise. Following clustering based on chromatic and achromatic thresholds, see methods. Every pixel in every image is assigned to a categorical color identity. The total number of colors in an image provides a measure of richness, and the numbers equivalent of the Shannon diversity of the colors, taking into account the relative area covered by each class of colors, provides a measure of color diversity. Individuals with higher richness scores have more colors, and individuals with more colors, more evenly distributed in terms of their relative areas, have higher diversity scores. S6 figure, accumulation of unique notes plateaus at time windows of approximately 10 seconds for most species. S7 figure, pairwise comparisons of acoustic richness, number of unique note types, estimates for windows between 5 and 50 seconds in duration.